Our understanding of genetics is increasing all the time. But could a new process called gene editing be the answer to some of the biggest problems facing farmers? Barley and potato are Ireland's most important crops. In 2016, we produced nearly 1.5 million tonnes of barley, and our annual national spend on potatoes is a whopping €162 million. But crop disease remains one of the biggest threats to our agricultural industry. David Walsh, chemist, runs his family's farm in County Laois.
For me, barley is important because where I am is the real malting barley heart of Ireland. All my neighbours, all my friends are all growing malting barley. It's going into beer, it's going into whiskey, and then we'll be growing feed barley as well, which ends up in the food chain for animals. So
it's an intrinsic part of Irish food, really?
It's a very important part of Irish food. I mean, obviously, beer and whiskey production is very important for us in this country. A lot of the animal feed that is being produced is made up of a ration that includes a lot of barley in it, so it's very important to keep the whole industry going.
And tell me about the challenges of being an Irish barley farmer.
Well, the good thing about growing barley in Ireland is that it rains a lot because that helps the yield, but the bad thing about being an Irish barley farmer is that it rains a lot. So, you know, we're getting a lot of disease pressure in barley. Keeping disease out of the crop is the most important thing that we need to do as barley farmers.
Disease control and crop loss in potato and barley costs Irish farmers an estimated €63 million per year. And I'm meeting Teagasc researcher Ewen Mullins, who is investigating new ways to tackle the problem using genetics.
We have a huge problem constantly trying to battle diseases in our crops. Each year, potato growers would spray maybe once a week, up to 12 or 13 sprays to get their crop out of the ground. Wheat growers, 3 to 4 sprays, and one of the diseases in wheat called septoria is becoming very difficult to control. In addition, barley, while we have some resistant varieties, we also have diseases there such as rhynchosporium, as we see here on the leaf.
This looks like an insignificant piece of grass, but this is pure science.
It is, because what we're trying to do is we're trying to develop varieties that resist these diseases.
Some people may have concerns about advanced plant breeding techniques, but we've actually been doing this for millennia, crossbreeding plants to improve their characteristics and resistance to disease. For decades, Teagasc have been at the forefront of this genomic research to identify and effectively breed out the genes in crops that are susceptible to disease.
You can develop resistant varieties, okay? And one of the things is that when you develop a resistant variety, you have diseases such as this guy here, rhynchosporium, which can evolve quite quickly and overtake that resistance. So it's a constant arms race, in effect. You develop a new variety, the disease overcomes it, you develop a new variety, and so forth. We want to try and break that cycle. We want to try something new. And what we've been doing is looking at what we call these susceptibility genes. Basically, the genes that you would put into a variety to make it resistant are what we call resistant genes, and they're effectively produce an antibody. But what happens is when the disease overcomes that resistance gene, breaks it down, in effect, that gene becomes a susceptibility gene. What we're doing now with editing is if you switch that useless gene and you reprogram it, in effect, reboot it, what you're going to do is you're going to reverse the time clock on evolution.
Wow, that's amazing.
It is amazing.
It's just a reset button.
And you can only do it through editing because all you have to do is change one single base change in the DNA.
Wow.
So this is basically plant tissue cultures.
Right.
So these happen to be potato plants, but to start genome editing, you need to have sterile, really healthy plant tissues, and that's what these guys are. So we put a lot of effort into making sure our cultures are clean, they get perfect light, no diseases, and they sit in this lovely jelly down here, which is basically just sugar and nutrients, and they're happy out.
This is a prime example.
If you're a plant, this is where you want to be.
Okay. The concept behind gene editing is not new, but now a pioneering technique called CRISPR-Cas9 has been developed that's cheaper, more accurate and much faster than current methods. It's predicted to be a game changer, but of course, it's not without controversy.
There are a huge number of applications for this technology, and I think that's why so many people are excited about it. You know, there are studies going on at the moment, for example, to edit mosquitoes so to edit their genome so we don't have transmission of malaria. That would be a massive thing. You're looking at studies actually trying to edit out allergens in peanuts, so you don't have children going into anaphylactic shock who have a peanut allergy. There has been a huge ethical debate around gene editing. Most of it has actually been in relation to editing of the DNA of the human embryo. And that's because if you actually make a change in the DNA of the human embryo, that's passed on to subsequent generations. I suppose what's really welcome actually with gene editing, unlike a lot of other new technologies we've seen in the last 20 years, is that there is this ethical discussion that has started really early. And that's not always been the case, because usually the old adage is that ethics is always chasing science. So by having this debate early, we raise awareness, as this program is, about these kind of issues, and that allows us to have a kind of more democratic debate about what we want to use these technologies for, where people think the limits should be placed, because I don't think that's a question simply for scientists or simply for governments, but that's a question for us as a society about what we think is acceptable or not.
The ethics debate will continue, but for crops, Teagasc are keen to start the discussion by exploring the opportunities and challenges of genetic editing technologies.
And when will farmers start using gene-edited barley in their fields?
So within Europe, we're having discussions at a policy level about editing and these modern biotechnologies and how they can apply to food production. So hopefully there will be no policy restrictions on these, and then this is just normal conventional breeding. In terms of setting up, we would hope to have edited material in the next 12 to 18 months that would then go into the glasshouse behind us, that we can then start to screen it for diseases and anything else. And after that, then we'll decide whether it's worth going into the field.
I think anything that is going to provide us with better quality feed at the end of the day is worth looking into. The thing that I would have to look at is what is the consumer perception going to be. So there's no point in me using, say, a gene-edited barley if my consumers are going to say, well, I'm not drinking beer that's made from gene-edited barley. So it's important for me that consumers are happy with it, and if they are happy with it, then absolutely, why not?